Okay, so today we have Viridiana Martinez. She's a graduate student in ecology and conservation biology. And today she'll be talking to us about stress hormones and blood parasites.
All right, well, thanks for the introduction. So as Michael said, today I'll be introducing my research, investigating these interactions between stress hormones. So to start, I want to take a moment and discuss disease refugia, and that's a concept that occurs across multiple taxa around the world. So disease refugia is an area within a host range that is absent from disease, and this has been found to occur in many organisms around the world. Mosquito, flea, and bacterial diversity decreases with latitude and altitude. And one of the oldest observations of this spatial pattern has been observed with human and avian malaria. A very famous case study investigating disease refugia occurred in Hawaii. So after the introduction of the southern house mosquito, populations of endemic bird species like the Hawaiian honeycreeper were decimated by avian malaria. So avian malaria is an intracellular protozoan parasite that is transmitted by mosquitoes. And it was found that only at higher elevations where these endemic species were found to be free from disease. It was also found that the cooler climates at these higher elevations slowed the development rates of both the mosquito and the parasite, resulting in areas that are disease-free. And this phenomenon has been observed across different species and pathogens. A separate example is the shift in the distribution of fungal pathogens in boreal toads. Boreal toads in southern Rocky Mountains have lower extinction risk at higher elevations from fungal pathogens that cause disease. And other potential factors that may contribute to elevation disease refugia have been studied in the immune response. So one example of this is the immune response of the Turkish blind mole rat. It has been found to have a greater immune response at higher elevations than at lower elevations. Elevation gradients occur along montane regions. As elevation increases, temperature decreases. This temperature gradient can potentially be a factor for acute climatic shifts at higher elevations, potentially affecting the homeostasis of resident organisms, causing stress. These montane regions are important areas to study, especially with the current changes in climate. Bird habitats and breeding ranges have been observed to move upward in elevation as climate continues to change. And for high elevation species, this may not be an option, increasing their conservation concern. So how these birds respond to environmental differences at higher elevations compared to their usual home ranges are crucial for future conservation efforts. One way to measure the response to environmental differences is by monitoring circulating stress hormone levels. This led to our research objectives investigating the effects of high elevations on multiple factors in bird health. The first objective is to determine the changes in avian malaria dispersal along elevation gradients. The second objective is to investigate the influence of high elevation environments on corticosterone levels. And corticosterone is an avian hormone that is released in response to external stressors. And third, our objective is to identify patterns of avian malaria in relation to corticosterone levels along elevation gradients. We hypothesize that birds at higher elevations will have decreased levels of avian malaria, and at higher elevations will also have higher levels of corticosterone. We hypothesize an inverse relationship between avian malaria and corticosterone at higher and lower elevations. But today I will only present on the influence of high elevations on corticosterone levels. Stress is a perceived or actual threat to homeostasis, and stress results in a hormonal response to adjust homeostasis. External stressors such as high or low temperatures, hunger, or predation events register in the brain and result in a cascade of events leading to the release of hormones. The hormone that is released in response to a stressor in birds is called corticosterone. This hormone then affects the behavior and physiology of the individual. Corticosterone is important in influencing the fight or flight response after experiencing a stressor, changing the behavior of the individual, as well as affecting the physiology of birds, influencing their immune response. For example, acute levels of corticosterone are known to have immuno-enhancing effects, while chronic levels are known to have immunosuppressive effects. Corticosterone can be analyzed from both serum and feather samples. Serum samples provide a snapshot of corticosterone levels at that specific moment in time, while feather samples provide historical levels of stress, as corticosterone is diffused into the feather during development, which occurs over the course of a few days to a few weeks. To investigate how corticosterone levels change along elevation gradients, we sampled across various elevations in West Texas. We have 3 different elevation gradients ranging from low to high. At low elevation, we sampled at Black Gap Wildlife Management Area, which ranged from 2,000 to 4,000 feet. At medium elevation, we sampled at Elephant Mountain Wildlife Management Area, as well as Sierra Diablo Wildlife Management Area that ranged from 4,000 to 6,000 feet. And at high elevation, we sampled at the Davis Mountains Preserve and the Guadalupe Mountains National Park at 6,000 to 8,000 feet. And we conducted this research by catching birds via mist nets and obtaining blood and feather samples. Mist netting is a common technique that is used by avian researchers to capture wild birds during flight. The birds are quickly removed from the net once they hit the net, and we quickly take blood and feather samples. We take blood samples by collecting blood volume that is less than 1% of the body weight of that individual from the brachial wing vein, as well as taking 2 central tail feathers. And the reason why we collect the 2 central tail feathers is because they're known to regrow quickly, have minimal, if any, adverse effects to flight, and is a very common practice among avian research. Analysis of the blood and feather samples for corticosterone was conducted via enzyme-linked immunosorbent assays, also known as ELISAs. And ELISAs use immunofluorescent antibodies that bind to the corticosterone in the sample to determine the concentration of corticosterone. An analysis of the blood samples for presence of avian malaria will be conducted via polymerase chain reactions. This is a table demonstrating the number of individuals of one species caught at different elevations. Here we analyze samples from ash-throated flycatchers from low to medium elevations, western tanagers from medium and high elevations, and Scott's orioles, cactus wrens, northern mockingbirds, and paraloxia at low and medium elevation ranges. Although the total number of individuals for this analysis is 45, we only compared individuals of the same species sampled at different elevations. This is preliminary data from feather corticosterone analysis. The x-axis represents different species that were caught at low, medium, and high elevations, and the y-axis represents the average corticosterone concentration in picograms per millimeter. These birds are also grouped by gill. Ash-throated flycatchers, western tanagers, and Scott's orioles are all species that primarily feed on insects during the breeding season. Cactus wrens and northern mockingbirds are generalist feeders, and paraloxia primarily feed on seeds. With the exception of the western tanager and northern mockingbird, there seems to be a general trend of higher levels of corticosterone at lower elevations. This is pretty interesting because our preliminary data does not support our hypothesis that birds caught at higher elevation would have higher levels of corticosterone. However, it should be noted that not all of our samples have been analyzed, therefore substantial conclusions cannot be drawn at this time. Currently, sample sizes comparing our target species along elevations are low, and this may be affecting our average corticosterone levels. Again, this is ongoing research and not all of our samples have been analyzed yet. Sampling during the breeding season may also be influencing corticosterone levels. Corticosterone is usually lower during the breeding season than at any other time because the response to external stressors is usually suppressed to allow for breeding success. Since higher elevations tend to have cooler temperatures, it is possible that heat stress may be a contributing factor to corresponding stress levels. Higher temperatures may affect food availability and increase food provisioning during the breeding season, which may also increase corticosterone. As I've said previously, this is an ongoing project, so we will have one more field season of sampling to increase our sample sizes of target species found at various elevations. Sampling at all the field sites will be equally distributed, spending similar amounts of time at each of the sample sites throughout the breeding season. And we will also complete our parasite and corticosterone analysis to look at patterns of parasite prevalence along elevations, as well as determine any potential relationship between corticosterone, avian malaria, and elevation. I would like to acknowledge everyone who made this research possible: my advisor, my committee members, my colleagues and field assistants. And I'd like to thank the entire Grace Lab for their continued support throughout this project, and especially thank my funding sources, the Applied Biodiversity Science Program at Texas A&M University, as well as the Schubot Center for Avian Health. And questions?
Our next speaker is Maureen Hayden. Maureen is a graduate student in biology at Texas A&M, and today she's going to be talking about microplastic pollution. So Maureen, I'll hand it off to you. Feel free to share your screen, unmute yourself, whatever you need to do. You are muted at the moment.
All right, can everybody hear me?
Yes, yep.
Awesome. Can everybody see my screen?
I think so, yes.
Okay. Do you want me to wait until 10:40, or would you like me to start now?
That's totally up to you. If you'd like to wait, if you have people you expect coming in at 10:40, that's fine. We can kill a minute here. If not, just feel free to go ahead and jump in.
Okay. All right, I will get started then. So when I first came to A&M, I didn't imagine I would be doing plastics research. Actually, I didn't think I would really be as involved in this movement as I am. I first became aware of the plastics problem during my freshman year of undergrad when we were assigned our summer reading project. We hadn't even gotten to college yet, and they had us read a book. It was called No Impact Man by Colin Bevan, about a man in New York City with his family who attempts to go completely green for an entire year. No public transportation, no electricity, only buying local produce with no packaging, and here's the kicker: no toilet paper. So for all of us who are stressing out about the empty shelves, it's okay. Take this book, No Impact Man by Colin Bevan, give it a read. We can go with less toilet paper than we think we need. But after reading this book, it really got me thinking about how my own actions contribute to our consumer market and plastic waste. And then as an aspiring marine biologist, I really wanted to work on focusing on anthropogenic impacts on our coastal environments, which has led us to our topic today of microplastic pollution on the Texas coast. So microplastics are defined by the National Oceanographic and Atmospheric Administration as anything that is 5 millimeters in length, or about half a centimeter. So that's about half the width of your pinky finger, the size of a popcorn kernel, or about the size of a pencil eraser on one of those like #2 Ticonderoga pencils. Or just think of a bag of frozen peas and then go and throw that all over the beach. And that's what we're doing to our environment, except instead of frozen peas, it's microplastics. So the goal of this study was to determine the extent of microplastic pollution on 3 Texas state park beaches, and to try to get some preliminary data on just how bad the problem is. So for my study, I'm working with Searim State Park, Galveston Island State Park, and Mustang Island State Park. I chose these sites because they give me a representative coverage of the Texas coast. And then additionally, all of these parks have areas that are cut off to vehicle traffic, which allows me to get a consistent transect each time while also eliminating variables from one anthropogenic factor, which would be the cars. So for our sampling collections, we've been going out seasonally, fall, winter, spring, and summer, although we may not get spring data at this point. We usually will run a transect parallel to the shore at the high tide line, and this transect will be 50 by 1 meters. The way we came up with this number is it's a modified protocol of the NOAA Marine Debris Monitoring Program. The original protocol called for a 100-meter transect, and we didn't have the time or the resources to do that. Then what we would do is we would collect small marine debris, as we are calling them for this survey, and that was anything that would fit into a 50 milliliter centrifuge tube that would fall across our 50 meter by 1 meter transect. After that point, we would collect these debris, and then we would take them back to the lab, and we would catalog them and visually observe them. So we would look at them under the scope, we would take photos of each one, and then what we would do is we would characterize them according to a couple of things. One is it a primary or a secondary microplastic? So is it a plastic particle or a fiber? Those would be secondary plastic, something that breaks down from something that's larger. Or is it a primary microplastic? And these are usually called nurdles. So they're the first step in the plastic manufacturing process. They're plastic pellets that are melted down to create things that you and I consume as plastic products. And so those are small plastic particles that are intentionally created to be small. Next, we would categorize them by size. So we would measure them using a program called Fiji or ImageJ. Then we would also take note of their color, and then we would categorize them into micro or macro plastic. And this is just based on whether or not they fit that NOAA definition cutoff of 5 millimeters greater than or less than. So here's an example of some of our categories. So in the upper left-hand corner, we have a microplastic, which is less than 5 millimeters in blue. In the upper right-hand corner, we have a macroplastic. In the lower left-hand corner, we have a green macro fiber. And then there's an example of those nurdles I was telling you about. They're usually between 3 millimeters to 5 millimeters in length. And they're very consistent in size and shape. So when we look at the data and we compare our summer and fall samples at all 3 of our state park sites, we found that there were consistently more plastics than nurdles at all 3 of our sample sites. And you can see that between our dark blue and our lightest blue color here on the box and whisker plot. And then when it comes to the difference between plastics and fibers, we found that there was a significant difference between Mustang Island State Park and Searim State Park. And then if we look at microplastics specifically, so this was only plastics and nurdles category that fell below that 5 millimeter cutoff line. We saw that there was a significant difference between Galveston Island State Park and Mustang Island State Park. So that's that darker blue line. But we didn't see a significant difference between Searim State Park and any of our other sample sites when we combine the summer and fall data. So in the future, we want to look at the difference between sample sites and variation in marine debris within sample sites. We also want to continue sampling throughout seasons. So currently we were working on analyzing our winter samples from January to March, and we were hoping to go sampling this spring. We'll see what happens before the May cutoff for that. I also want to look at comparing sample methods. So the method I talked to you about today was doing a surface area transect, but I also want to look at doing methodology with sediment cores and sampling by volume to see if there's a potential bias in the way in which we sample for microplastics. And then I'm also hoping to do chemical analysis via either Fourier transform infrared spectroscopy or Raman spectroscopy to do materials characterization on those plastics that I collect. So for example, are we finding more polypropylene, more polyethylene, polystyrene, high density polyethylene? What is the source of those plastics that we're finding on the beach? So in conclusion, we found microplastics at all 3 of our sample sites during all of our sample seasons that consisted of both primary microplastics, so ones that are created intentionally to be small or intentionally to be of that microplastic size, and secondary microplastics. And we hope to continue this sampling protocol throughout the different sampling seasons, and hopefully for a whole another year so we can get comparative data. And with that, I would like to thank my advisor, my research committee, my lab mates, and the many undergraduate research assistants who have helped with this data. And in addition, I would like to thank all of my funding sources and my collaboration with Texas Parks and Wildlife. Are there any questions? And Michael, I would like to ask you to help with the chat box, if you wouldn't mind help reading the questions.
Definitely. Yeah, I'll keep an eye out. So thank you, Maureen. That was an excellent talk. That was very cool. So as we wait for people to type away or ask questions, again, if anyone has a question, you can feel free to unmute yourself and ask directly, whatever you're more comfortable with. So I'll go ahead and kick off the question. I'm not informed about many things, but so I'm wondering the nurdles you found. I mean, you don't find those as often as the other types of microplastics. Is that just because nurdles are not in like final consumer products very often, or is there another reason why they're not found very often?
So I'm really interested in this myself. There's actually a citizen science project called Nurdle Patrol on Facebook. If anybody wants to get involved, anytime you go to the beach when shelter in place is over, you do a 10-minute survey at the high tide line and you collect nurdles, and then you can mail them to the Mission Aransas Marine Reserve or just take a picture on Facebook. And they have found millions of nurdles, like, I mean, millions throughout since August 2018. So I'm wondering if maybe all the citizen scientists aren't collecting them maybe. Also, I've seen on the Nurdle Patrol maps that a lot of them are a lot closer to river and lake outflows than beaches necessarily, because that's where the plastic manufacturing plants are.
I see. Okay. Oh, interesting. Cool. Okay, so we have actually 3 questions here that have popped up. So first, do you have any estimates as to why there was so much more plastic pollution at Galveston?
I'm thinking partly because of tourist activity. I don't have the logs yet, but I'm hoping to work with the park rangers to get an estimate of like park usage to see if there is more tourist activity at Galveston compared to the other 2 parks. And then also Searim State Park and Mustang Island State Park are kind of in more quote remote locations, whereas Galveston Island State Park directly borders the seawall. So you're right up against all of that like city area and population coming from other sources.
Interesting. The next question is, what's your hypothesis on why there's a difference between sites?
So part of it has to do with, I'm trying to figure out, is some of it due to tourism or is some of it due to Texas? And part of it, like the Gulf Stream currents. So one thing that's really interesting about the Gulf is that we do not have steady currents like the Atlantic or the Pacific. We have a bunch of little loop currents, which could lead to potentially different trends in plastic pollution than what we're seeing on the East Coast or the West Coast. And what I'm thinking is that we might be seeing more consistent deposition of plastics throughout the year compared to what I've seen in the literature with other beaches where you find, like on the Northeast and the Northwest, where you find like less plastic in the winter and like a lot more in the summer. I'm thinking because of all of the loop currents, we might be seeing different trends in the site. And then also with Searim State Park, it's a lot closer to the Mississippi Delta. So I'm not sure if that has anything to do with like the difference in the plastic pollution. But I haven't seen a lot of studies on variation between beaches. A lot of the studies I've read, they just look at one beach only, and usually only at one time period.
Interesting. Great, thanks. And then we have one more question. Hi, Maureen. How were transect locations selected along the beach?
So I selected transect locations at high tide line, partially haphazardly. I had to choose some kind of a landmark to kind of help me. So usually what I did is we walked about like 100 meters away from the entrance, so that we weren't near like where a lot of the people traffic was. And then if you've ever been out to the beaches, there are the big like 100-gallon like trash drums. So we use those as our markers. And then we just pick the high, we just start there with our transects and we sample at the same spot every time.
Interesting. Thanks.
Hey, Maureen, I have a question. So I think it'd be nice to think about like what are the biological implications of plastics on the beach, right? So we know that we've got a lot of plastics, but do you have an idea of how it's going to impact different organisms and how it might impact them differently?
Yeah, that's a great question, Zach. I didn't talk about that in this PowerPoint specifically, but the other chapters of my dissertation are going to be focusing on feeding behavior in amphipods on the beach and how ingestion of microplastics might be affecting their survival. So past studies have found that invertebrates or even charismatic megafauna that have ingested plastics leads to not only just aggregation in the gut or just the presence of plastic in the gut, but it can lead to retention where those plastics will accumulate. It could lead to chemical transfer of pollutants into the gut of animals. Additionally, one big concern, especially with a lot of the amphipods on the beach, is that we're thinking it could lead to dietary transfer to higher trophic levels. So if small amphipods are eating the plastics and then a bird eats an amphipod that's eating a plastic, then you're not only getting transfer of plastics like directly if the birds are eating it, but you're getting that higher level trophic transfer. So stay tuned for next year on an update.
We definitely will, for sure. Thanks, Zach, for the question. Does anyone else have any questions for Maureen? Well, if there's no more questions, again, thank you, Maureen. That was very cool, very interesting, and like you said, we'll stay tuned. So everyone, come back next year, and we'll get the second chapter. Thank you. So we have about 5 minutes until we move on to our final presentation. We'll probably get there a little bit ahead of schedule. Before we get there, I just want to make a couple of quick announcements. So we have, if you've registered, unfortunately registration is closed at this point, but if you registered yesterday for our lunch with the speakers, that starts at 11:30, so pretty quickly after this session ends. If you did register for that, again, it's about an hour-long lunch where you can interact with our plenary speakers from yesterday. So that should be pretty fun. Other than that, we have our afternoon sessions. They start at 2:30 and they go until about 3:45. And then our final event of the day is we have awards and kind of closing remarks at 4:30. So come back for that. We offer an award for best presentation for each of our sessions. So it should be exciting. And then the other thing, actually, after lunch, so from 1 to 2:30, we have a virtual poster session. So for anyone who has a poster presentation, which you can find, again, the whole list of poster presentations on our website, we have a single page where you can just kind of virtually browse all of the posters and be able to read all of them. And we've asked all of the presenters of the posters to be just readily available by email. So their email is posted with their poster. So if you have any questions, you can email them directly. Again, not an ideal situation, but given the circumstances, hopefully you can still be able to do some pretty cool research that we have in the poster presentation. Okay, so again, we're a bit ahead of schedule, but I guess that's a good thing. We haven't had too many difficulties yet. So if it's okay, I'd like to go ahead and move on to our final presentation, Morgan Thompson. Hi, Morgan. So Morgan is a graduate student in entomology at Texas A&M. And today she'll be talking about insect herbivores interacting with something. I'll let her describe it. Thanks,
Okay.
Morgan. Take it away.
All right, great. So everybody can hear me?
Yes.
Yeah. Okay, great. Awesome. All right. Well, thank you all so much for virtually attending my presentation today. I'm really excited to share some research that I've been conducting over the past few months in the Helms Lab. And so I kind of want to begin my talk by just broadly stating that plants are under a constant threat of attack from a variety of antagonists. And so we often think about plants potentially as passive actors in these interactions, but in reality, plants have really complicated and sophisticated mechanisms for defending themselves against different types of antagonists. And so today I want to think and talk with you all about how plants defend themselves specifically against insect herbivores. And as insect ecologists, we typically think about plant defense against herbivores and break this down into two broad categories. The first being direct defense against herbivores. And so this is when the plant is producing a toxin or a physical structure, such as a trichome or thorns, that directly deters herbivore feeding on that particular plant. And following insect herbivory, plants also emit what are called volatile compounds or volatile emissions, as it's labeled on this figure. And these compounds are released into the atmosphere and then can be detected by other organisms, such as other herbivores, and they can actually serve also as a direct defense against foraging insect herbivores, or also serve as cues for those herbivores about the competitors on a particular host plant, as well as the location of a host plant. And then interestingly enough, it's also been demonstrated that these volatile emissions produced by plants that are damaged by insect herbivores can also actually attract predators and parasitoids. And so this attraction of predators and parasitoids to predate on those insect herbivores that are feeding on plants is referred to as indirect defense of plants. And so one other thing that I want to make note of is that these direct and indirect defense mechanisms that plants use to defend against insect herbivores are often mediated and regulated by signaling molecules, which we also refer to as phytohormones in plants. And so today I'm going to be talking a little bit later on about jasmonic acid. So this is one example of a signaling molecule that's produced endogenously in plants that helps these plants coordinate these different defense mechanisms against a particular insect herbivore. And so we know a lot about how plants defend directly and indirectly against a variety of different herbivore species, and we know how these interactions occur across a variety of different plant species as well. But what we often overlook in terms of thinking about these interactions is what's happening below ground, and how below ground herbivores could be contributing to these above ground interactions. And so in recent years, there's been some really interesting research that's come to light about plants that are attacked by root herbivores and the induction of increased resistance to above ground herbivores. And in particular, there's been a lot of research into the mechanisms of how this happens in corn plants, as well as different brassica species. But then this idea of root herbivory changing the plant defenses above ground also begs the question as to why plants would do this. Why would a plant change its defenses above ground when it's being attacked below ground? What could be the potential reasons for why we observe this root herbivore-induced shoot resistance? And so some proposed ideas about why this potentially could be occurring in these plants would be that first, this is a plant physiological constraint. So these plants, if they're attacked below ground, are responding systemically throughout the entire plant. And this is just a response of the entire plant to some kind of threat. The other idea is that potentially this is the herbivores manipulating plant defenses for their own benefit, i.e. they're changing the defensive status of the plants above ground to deter potential competitors that could be eating the plant or consuming plant material above ground. And then finally, this could also be a signal for plants of an increased likelihood of above ground herbivory. So if the plants are being fed on below ground, perhaps there's heavy herbivore pressure in their particular environment, and therefore their above ground defenses. But one thing that's really interesting to note about these previous studies in corn and brassica species is that most of these studies have been conducted with two different herbivore species. So one species that feeds below ground, and then a different species of herbivore that feeds above ground. And so for my research that I've been conducting in the Helms lab, I've been working with striped cucumber beetles. And so these are specialist herbivores of cucurbits, and the adults, as you can see here, feed on the above ground foliage of cucurbits, such as squash, which is the plant that I've been working with for my research, but they will happily feed on many different cucurbit species, such as cucumbers, pumpkins, watermelon, other gourds, etc. And then the larvae of these beetles actually feed on the roots of the plants. So the life stages of the beetle are separated spatially between feeding below ground and above ground. And we know very little about how the plant is perceiving the herbivory below ground, and then how this subsequently changes defenses above ground. And so that was really the overall goal of my project, was to determine the relative importance of root herbivore-induced shoot resistance in interactions between striped cucumber beetles and squash. And then again, I was thinking about these three non-mutually exclusive hypotheses of thinking about how plant physiology and the constraints of plant physiology factor into these interactions, if this could potentially be larvae that are controlling this interaction, and then finally, if below ground herbivory signals to the plant an increased likelihood of above ground herbivory, and then subsequently how the plants respond and adjust to this. And so then my specific objectives that I tested across a variety of experiments that I want to share with you all today were first thinking about if below ground herbivory affects the above ground production of plant volatiles. So as I mentioned earlier, plant volatiles are really important signaling molecules in above ground plant insect interactions. And then second, looking at if plant systemic phytohormone levels change as a result of below ground herbivory. So again, do these defense signaling molecules in the above ground portion of the plant change as a result of below ground herbivory? And then finally, thinking about if we can see changes in above ground insect herbivore behavior as a result of below ground herbivory. And so to think about that first objective, what we did was we set up above ground plant volatile collections. And so here we had two groups of plants. The first group of plants were larvae damaged. So we allowed the larvae to feed on the plants for 48 hours prior to above ground volatile collections. And then we compared these collections to a group of control plants. And so today I want to present the results for one specific volatile compound that we found to be of interest in this study. And so here I'm presenting the results for above ground volatile emission of beta-ocimene, which you can see pictured here. And then on the y-axis here, I'm presenting the volatile abundance. So this is the nanograms of beta-ocimene per gram of above ground plant tissue. And then on the x-axis on the left, we have our control plants, and on the right, we have the larvae damaged plants. And what you can see is that in the larvae damaged plants, we see a significant increase in the amount of beta-ocimene that the plants are emitting above ground. And so this was a really interesting finding to us, to see the volatile profile change significantly in response to this below ground damage. And so we wanted to follow this up by looking at systemic plant phytohormone levels. And so again here, we were comparing control and larvae damaged plants. And so for this particular experiment, we allowed the larvae to feed for either 24 or 72 hours. And so here pictured on the left, you can see a petri dish containing some of these larvae that we would place on the roots of plants and then compare those larvae damaged plants again to the controls. And here we collected leaf tissue and analyzed these for phytohormones. And today specifically, I'm going to present the results for jasmonic acid, which, as I mentioned earlier, is a defense signaling molecule or a phytohormone that the plants use to coordinate defenses. And we measured jasmonic acid as a proxy for plant defense to give us an idea of how the plant is beginning to coordinate its defenses against an herbivore. And in particular, jasmonic acid is often measured for chewing insect herbivores, such as the beetle that I've been working with. And so here I'm presenting the results for total jasmonic acid levels on the y-axis. So this is nanograms of JA per gram of leaf tissue. And then on the x-axis, again, on the left, we have the control plants and the right, the larvae damaged plants. And so here after 24 hours, what you can see is that we detected no differences in the above ground jasmonic acid levels between control and larvae damaged plants. And then when we look at 72 hours, so this is after 72 hours of below ground herbivory or larvae damage, we see, again, no differences between control and larvae damaged plants. And so despite this lack of difference in our phytohormone levels, we were interested in still if this difference in the volatiles that we detected could mediate interactions and behavior of the above ground adult beetles. And so what we did to test this was we set up choice tests for adult female beetles. So here we had, again, comparing our control versus larvae damaged plants. And so I'm showing here a picture of the entire setup that we had, and we blocked the visual cues with pieces of cardboard in between our choice arenas. And then within each choice arena, we had two different plants. One that was a control plant with no larvae, and then a larvae damaged plant. And we allowed those larvae to feed on those plants for 48 hours. And then following that 48-hour mark of herbivory, we added 6 starved female adult beetles to each of these cages, and we allowed those female beetles to feed for 48 hours. And then following 48 hours of feeding damage by the adult female beetles, we quantified the leaf area of feeding damage using image J. And so then here I'm presenting the results from that experiment. So on the y-axis, we have the leaf area damaged in centimeters squared by the adult female beetles. And then again, on our x-axis, the control and larvae damaged plants. And we can see here that the adult female beetles damaged the control plants significantly more than the larvae damaged plants. So they fed on these plants significantly more than those larvae damaged plants. And so then one other interesting piece of data that I want to share with you all from this experiment was when I broke down the experiment at the very end, I also recorded data on where I found the adult female beetles at the very end of the experiment. So which plants they were either resting on or actively feeding on at the very end of the experiment. And so here I'm presenting what I'm calling adult female beetle settling behavior, so which plants they chose to settle on by the end of the 48-hour period. And so on the y-axis, I'm presenting number of beetles. And so again, there were 6 beetles per choice arena. And then on the x-axis, the control versus the larvae damaged plants. And we can see on average that the beetles significantly preferred settling on the control plants relative to the larvae damaged plants. And so in conclusion, if we think back to those initial objectives that I had, I was really interested in testing if below ground herbivory affects above ground plant volatile production. And what I found was that yes, there are increased levels of beta-ocimene emission in plants that are damaged below ground by larvae. But interestingly enough, we didn't find any evidence for changes in systemic plant phytohormone levels. So jasmonic acid or JA levels did not change in the leaves of the plants that were damaged by the larvae below ground. And then finally, when we looked at the adult feeding behavior, we found that yes, adult female beetles consumed less leaf tissue and avoided settling on plants that were damaged by the larvae. And so we have a few ideas about what could potentially be going on in this system. So first, we think that beta-ocimene could be directly repelling adult female beetles. And this could be for a couple of reasons. And again, thinking back to all of these potential 3 factors that may play a role in how plants are responding to below ground herbivory above ground, we think that potentially the adults could be detecting these chemical cues and then avoiding competition with their offspring. Or we think that potentially these larvae could be manipulating plant defenses to sequester a greater number of resources from the plants for themselves. But then also we have to consider that this could be the plant and how the plant is perceiving both the below ground antagonists and then the potential threat from above ground antagonists. And so we think that potentially the larvae damage below ground could be mediating volatile defenses above ground for the plants. And so again, as I mentioned earlier, these volatiles could be playing a role in direct or indirect defense for the plants. And then finally, we have yet to tease apart the below ground defense mechanisms for squash plants. However, we do have some really interesting evidence for cucumber and how cucumber roots are responding to below ground herbivory. And we have some evidence right now that both direct and indirect defenses are playing a role in these interactions. And so stay tuned for a paper that will hopefully be coming out from the Helms Lab soon about some of those really cool interactions. And so with that, I would like to acknowledge my advisor, my lab, Texas A&M, the entomology department. And with that, I will take any questions. Thank you.
Thanks, Morgan. That was really great. That was really cool. Okay, so right away we have a question. So we have one that says, you only measured JA, jasmonic acid. Are you planning to measure other phytohormones? And if so, which one do you think could be playing a role?
Yeah, great question. Thank you, Lauren. So we did not only measure JA. So we have, and I have a few slides here. So we also have SA levels in the leaves of the plants right now. And so here I'm showing total salicylic acid. So this is nanograms of SA per grams of leaf tissue. And what we saw here was an interesting suppression of salicylic acid in the larvae damaged plants after 24 hours of feeding damage, but this seemed to disappear after 72 hours of feeding damage. And for folks who aren't super familiar with plant hormones, salicylic acid usually plays a role in plant defense against pathogens and piercing sucking insects. So this was another interesting finding to us. And so we're trying to do some work to follow up on this, but in terms of phytohormone levels, this is what we know as of now is jasmonic acid and SA. So yeah, great question. Thank you. Yeah.
We have one more. What about ABA and ethylene?
Yeah, yeah. So that would be really cool to get a handle on what's happening with abscisic acid and ethylene. So we haven't quantified ABA. That would definitely be another really interesting hormone to look at. And then ethylene is definitely really tough because it's a gaseous molecule. So it's hard, particularly when you're thinking about below ground levels and potentially how that would be moving from below to above ground to quantify ethylene. But yeah, we have some ideas to maybe look at that as well. We may look in the shoots of the plants. There's been some previous work about people who have been able to actually use a syringe to extract ethylene directly from the shoots of cucurbit plants. So you may think about that.
Interesting.
Yeah.
One more question. Do you think that the below ground volatiles phytohormones are playing a role in your system?
Yeah, great question. Thank you, Natalie. So yes, I definitely think that there could be some interesting interactions going on with below ground volatiles as well as phytohormones. So we haven't quantified any below ground volatiles for squash plants yet, but we do have some data on root phytohormone levels, if I can figure out how to share it. So we have jasmonic acid levels for our 24-hour and 72-hour mark as well. And here again, we're not seeing any significant differences, but potentially at that 72-hour mark, it does look like we're trending towards an increase in jasmonic acid levels. So that might be a future direction that we can go down. And then also kind of a similar trend for salicylic acid in the roots, where we're seeing that increase at 72 hours, but no significant differences yet. Yeah, that could indicate to me that potentially we need to just look at a time point beyond 72 hours to look at the phytohormone levels there, and see kind of what's happening and how the plants are responding. And I also know that from some studies with corn, there's definitely probably really interesting interactions going on with volatiles as well, and from some work in our lab too. So yeah, lots to do still for sure. And I think maybe there's another chat question, so thank you.
Yeah, go ahead and address it if you'd like.
Oh, okay, perfect. So it says, have you tried quantifying JA and SA levels in roots also? Yeah, so that was the data that I just showed here. So these are the root SA, and then for that previous slide, the root JA levels. So that's what we're seeing so far. Yeah.
Awesome.
Yeah, so any other questions from anybody?
I just have a small question.
Yeah, sure, go ahead.
Yep. So I'm just curious, have you tried the other way around, the effect of above ground herbivory induced resistance to the below ground thing? Yeah,
so that was my last slide of extra data that I included. So we haven't quantified anything in terms of phytohormones or volatiles and how they're changing as a result of above ground herbivory. Previous studies in other systems indicate that yes, likely above ground herbivory alone can probably change the volatiles as well as the phytohormone levels below ground. But when I did do a small study on the performance of larvae on plants that were damaged above ground by adults, what I found was that there were no differences in the mass gain of the larvae. So another way to say that is that the larvae happily fed on plants that were undamaged or damaged by adult beetles above ground. So that's our one piece of evidence right now, but I think that definitely would be an interesting route to continue to pursue. And I guess it's also of note that these are specialist herbivores that are feeding on these plants. So likely there are probably some potentially different interactions that are going on here than maybe you would see with a generalist insect herbivore as well.
Have you happened to take some phytohormone levels for this experiment also?
I did not know, unfortunately.
Okay.
Yeah. So I just the performance data, yeah.
Okay. Cool, thank you.
Yeah, thank you so much. Thanks for your questions.
Great. Well, unless there's any other questions in the last minute here. So I'd like to thank you, Morgan. Thanks for the wonderful presentation.
Thank you.
And thank you to all of the graduate presenters today. I think we had a really cool session. Okay, so that's officially the end of the general ecology session.